Sixth-year biology students sitting their leaving cert in June 2026, protein synthesis, the anabolic reaction, which involves transcription and translation, is something that causes confusion again and again. So we're going to break this down. In this video, we are going to look at all things transcription. So first of all, we're going to be talking about DNA. DNA, deoxyribonucleic acid. DNA, remember, is made up of different genes. Genes are sequences of DNA that will code for the production of a protein. However, remember, only 3% of DNA is coding for protein production. The other 97% of DNA is non-coding for protein. Now, DNA, as we've seen before, is a double helix. It's double-stranded. It is too large to fit out through the nuclear pores to get to the ribosome where proteins are made. So if we unravel or untwist our DNA for the sake of the kind of visual here, what you will see is that we have two separate strands of DNA. Now, one of these is going to act as our template strand. And we're going to copy our code from our template strand onto a single-stranded mRNA, M standing for messenger RNA. Then that messenger RNA is going to carry the code out through the nuclear pore and to the ribosome so that a protein can be made. So how does this happen? Well, imagine there's a gene that we want to copy, and we're going to say the gene that we want to copy is in between the two dashed lines. Just before the dashed line, we have an anabolic enzyme called RNA polymerase, and that's going to hold and join the mRNA nucleotides together as we make them. So we're only focusing on our lower template strand. Let's remember, first of all, our complementary base pairs. We are going to have a DNA template, and RNA is going to come in and bond to its complementary pair. So if DNA has a C exposed, a G will attach. If DNA has a G exposed, a C will attach of mRNA. If DNA has a T exposed, an A on mRNA will attach. And if DNA has an A exposed, a U from mRNA will attach. Okay, so we have A, U, G, C. And they're the complementary base pairs. So what happens is, just before the anabolic RNA polymerase enzyme, the two DNA strands will separate. They don't separate in big sections. They literally only separate above where the RNA polymerase is. So as you can see here, we have a C, T, A, G, C exposed. Okay, so what's going to happen is the C is exposed, so mRNA is going to start to form. The nucleotide G, guanine, is going to come in because there's a C exposed and its complementary base pair is going to be a G. Then we have a T exposed, so an A is going to come in, and the G and the A are going to be bonded together by the RNA polymerase. And this is going to continue up until the end of the gene that we want to copy on our template strand. And the RNA polymerase each time is rejoining those nucleotides together. Eventually, what's going to happen is we make our mRNA strand, and our DNA strand, the RNA polymerase, is going to join it together, and we're left with our mRNA. That mRNA is going to carry the code now out of the nucleus to the ribosome, where it is going to bond on in the ribosome at the start codon, and the tRNA is going to come in with the amino acids that it is coding for. So it is important to know that the mRNA is complementary to the DNA. So we have the DNA template, the mRNA is complementary to it, the RNA polymerase anabolic enzyme joins the mRNA together to form that mRNA that's going to carry the code out of the nucleus to the ribosome. And that process is known as transcription, getting the code from DNA onto a single-stranded mRNA using one of the DNA strands known as our template strand. So that is transcription, which is the first part of protein synthesis as part of the Unit 2 DNA and RNA chapter. The next video is going to be on translation, which comes next.